So what we're going to look at in this session is focusing on how microorganisms are used within predominantly the food industry as an application of biology. And a little bit like what we talked about last time, we've got a significant number of links and connections to different parts of the course. This is a really good opportunity for you to go back and embed the understanding and make those connections. So first of all, we've got to think about classification of organisms. And I've got the textbook reference there for you to have a look at. But the classification of organisms is first of all broken down into either being a prokaryotic type, which is essentially a bacteria, or a eukaryotic type, which is everything else. Now, the eukaryotes have got internal membranous structures. So we've got a nucleus, which is a true nucleus with a nuclear membrane, etc., etc., etc. Now, yeast is a type of fungus, which is a eukaryotic cell, but it's a unicellular organism. So yeast is a fungus, it's a eukaryote, and it's unicellular. And within yeast, you've got the characteristics of living things going on, and all of that metabolic processes are using enzymes to control the rate at which that reaction takes place. And again, we need to connect all of our knowledge about enzymes and how they work. We've got the active site being specific to the shape of the substrate. It's going to lower the activation energy, which allows those chemical reactions to take place. And again, page reference is there for you to look back at. Now, when we look at the production of bread, what you need to think about is the use of wheat flour. And wheat flour comes from a cereal crop, and the cereal grain contains both starch and it contains enzymes. We're going to mix wheat flour with a water solvent, and the yeast within that mixture is going to start to do anaerobic respiration. Now, remember what we know about anaerobic respiration. This is respiration without the use of oxygen. And in this case, with the yeast, when we break down the glucose, we're going to do that to produce ATP. And in so doing, we're going to produce the waste gas, carbon dioxide, and we're also going to produce alcohol or ethanol. So yeasts are eukaryotic cells, and when they're mixed with wheat flour, they've got access to starch. They've got enzymes which are going to break that starch down into maltose and then glucose. They're going to have that easily dissolved because you've got the presence of water, and they're going to undergo anaerobic respiration to produce alcohol and carbon dioxide. Here we've got the equations that we need to think about, and remembering that all of these are enzyme-controlled reactions. So when the yeast has got access to glucose, it's going to generate carbon dioxide and it's going to generate ethanol. Now, you might think, well, your bread's not alcoholic, but what will happen eventually is the alcohol will evaporate. The carbon dioxide, which is released from the yeast, is going to allow the bread to rise. So we've got that fluffy texture. Another thing that I want you to consider here is that the flour is connected to a grain, and the grain is germinating. So, and typically, barley seeds can be used also in the production of alcohol. And barley seeds are going to undergo germination, and they're going to break down starch into soluble products that can be used for the germination of that seed. If we look at this next slide here, this has been a question on a past paper quite a number of times, and what you need to think about here is how yeast is doing respiration anaerobically and producing alcohol. What we can see is the lime water, and remember that when carbon dioxide bubbles through lime water, it's going to undergo a change from clear to cloudy. The yeast and the mixture of sugar, glucose in this case, is in the bottom of that boiling tube, and we've got a layer of liquid paraffin. Now, the paraffin there is acting as a barrier to prevent any oxygen from getting to the yeast, which means that the yeast has to do anaerobic respiration as opposed to aerobic respiration. And what can also be appearing on a past paper question is, instead of having lime water in that test tube, we can have bicarbonate indicator. And bicarbonate indicator also tells us the rate of production of carbon dioxide. So what happens if you produce carbon dioxide is that will dissolve in the bicarbonate indicator solution, and that will make it acidic. And the more acidic it is, we get a color change. So it's neutral in this kind of orange region here. And as that yeast does anaerobic respiration, produces carbon dioxide, it'll be dissolved in the bicarbonate and it will be changing color, eventually becoming this yellow shade here. Then what we've got to think about is the application of this on an industrial scale. And what you can see in this image here is an industrial fermenter. And fermentation is anaerobic respiration for yeasts. And what we need to think about here is how industrially we control that process to maximize the product that's being produced. And we need to think about the substrate, that is, what type of sugar are we going to give to our yeasts? To the production of beer, it's typically barley, and barley is going to contain starch, it's also going to contain enzymes, and the enzymes within the barley are going to get broken down to release sugar. You add that to the yeast, so the yeast has now got access to sugar. If you give the yeast the appropriate temperature, the optimum temperature for its enzymes, it will react quickly. In a similar way, you want to have the optimum pH, which is necessary for the enzymes within the yeast to do anaerobic respiration. Now, these industrial vats are called aseptic. That means they are free of bacteria. And the process of making food where we minimize other contamination of bacteria is called pasteurization. So pasteurization is when you use mild temperatures to minimize the contamination of your beer production from other microorganisms. This diagram is very, very similar to the one that's in your textbook, and it's looking at all the things that are involved within the inside of that vat. And what we've got here is this idea of a cooling jacket, so that's controlling the temperature. We've got the control of movement of the substrate and the yeast by the use of those propellers. So take a good look at that diagram within your textbook. And the last thing that we need to consider is the production of yogurt. Now, when we get back to school, hopefully we'll have the chance to actually do this process ourselves. But here you can see what the process involves. This time we're using microorganisms, but we're using bacteria. We're using lactobacillus, and sometimes we mix the lactobacillus with something called streptococcus. Streptococcus can be pathogenic, but in this case it's not. And the bacteria which is used predominantly is the lactobacillus. And what happens here is that you've got sugars within milk, and the sugars within the milk are fermented by these lactic acid bacteria. And when they do that process, they will produce the sour tasting yogurt product. So what do? We take the milk, and first of all, we need to pasteurize it. We want to get rid of the microbes which are present naturally in raw milk, and we heat that milk up to about 85 degrees. The milk is then undergoing a process called homogenization. And what that does is it disperses the fat equally within the milk culture. Then what we're going to do is cool the milk right down to about 40 degrees, and we're going to add the bacteria. So the starter culture that is mentioned here is the mixture of the lactobacillus and the streptococcus. We then give that bacteria the opportunity to do anaerobic respiration. The protein within the milk is going to be digested into amino acids. Amino acids are acidic. The milk sugars are going to be fermented, i.e. it's going to go through anaerobic respiration, and that's also going to produce an acid, but this time it's lactic acid. So when the lactobacillus do anaerobic respiration, they do it in a very similar way to us. They don't produce ethanol, rather they produce lactic acid. Now, as the acid is built up, both the amino acids and the lactic acid, it starts to kill off the strains of lactobacillus which are in that mixture. So the numbers of the bacteria are going to fall. They don't fall entirely, which is why it's called live yogurt. And what we've got at the end of that is a thickened mixture. It's thickened because the protein starts to coagulate. It starts to coagulate because it's undergoing denaturation. Why is it going denaturation? Because of the drop in acidity. We can also then add all sorts of flavorings and colorings to that product.